Welcome to diagramming glycolysis, and this is glycolysis part 1. This all happens in the cytoplasm. So we're going to start with glucose, a 6-carbon compound. Now, in advanced classes, they care about more atoms. We're only going to care about the carbon, which are diagrammed as circles. So keep track of the circles as we go through this. We're going to start with our 6-carbon compound, glucose, and the first thing we're going to do is add some ATP to it. ATP creates a new molecule called fructose diphosphate, which is unstable and breaks apart and makes 2 pGals, or 2 3-carbon compounds. pGal's unstable, and it starts breaking down. The first thing that comes off are NADH, and then we get some energy out. We make 2 ATP. And the final product at this stage is pyruvate. Now, this all happens in the cytoplasm, and this is as far as bacteria go. They get a net of 2 molecules of ATP. 2 were used, 4 were made. So we have a net benefit of 2 ATP and 2 NADH. Now, the next step, we're going to continue with pyruvate. And we're going to break pyruvate down and eventually get into the mitochondria. So notice we went from a 3-carbon compound to a 2-carbon compound. We're going to abbreviate that 2-carbon compound as A because it stands for acetyl. Now the carbon dioxide comes off, so that's getting rid of our first 2 carbon dioxides. That gives us our C3 compound, pyruvate, to our 2-carbon compound. The next thing that happens is we're crossing the membrane to the inner matrix of the mitochondria, and we lose 2 NADH, and we get a helper. The only way we could get that 2-carbon compound across is with coenzyme A. So coenzyme A is going to help us get across that membrane to the matrix and finally making our molecule A-CoA. And this is the end of the intermediate reaction. So in reality, in a college class, they might talk about all of the enzymes. I think there's about 20 steps. We covered about 8 of those.